Missouri at the North American Mycological Association 4A 2022. We've been here for a couple of days. It's been a little dry, but we found a lot of cool mushrooms and a lot of biodiversity. So I'm going to take you guys into the ID hall and show you all the cool stuff we found. There's a lot of great mycologists wandering around. I can't predict who's going to be here, but I know that Alan Rockefeller's inside. I know we got some UV lights. Get a lot of cool mushrooms to show you. So come along. I'm going to show you what this is like. So I'm going to start you guys out with this big table that is a huge overview of fungal taxonomy. So taxonomy is the science of naming things. And down here we have these basal fungal lineages of early fungi that were kind of like aquatic parasitic things. And then we look at the evolution of fungi over time and the evolution of Ascomycota and Basidiomycota, which is the majority of fungi that actually form mushrooms. And from that, we had early things like jelly fungi and chanterelles, and we go all the way up the line to like the evolution of russulas and boletes and polypores and the whole span of all these mushrooms into Garicales, which is one of the biggest orders here of fungi. So it's pretty cool to have this big chart overview. We have a whole room here just full of mushroom nerds and mushroom samples. And I'm just going to take you guys through and kind of show you what's here. So down here, we're in phylum Basidiomycota. So these are mushrooms that look like the mushrooms you think of, right? They have cap, stem, gills, lots of polypores or basidiomycetes. So, like I said, it's been a little dry, but we have some interesting, these are Leucaria. I thought these are Cortinarius, but Leucaria. Tons of not honey mushrooms, these are jack-o'-lanterns. So this is Omphaliotis illudens. These are toxic, you can't eat them, but they do glow at night. You can actually dye fabrics with them too, which is pretty cool. Down here, we start to get into the honey mushrooms. So these are Desmalaria, and then these are Armillaria, Armillaria malaya, honey mushrooms. So these are parasitic white rot fungi that kill their tree hosts and then digest them. We've got some cool little like microscopic things here. I know somewhere around somebody found a cordyceps. That'd be an Ascomycete though, so that'll be on a different table. We have, ooh, wow. It's a really big piece of, I'm guessing this is Berkeley's polypore maybe? Let's see. What's cool is these all been identified by experts. And what we're doing in conjunction with NAMA, the organization hosting this, and Fundus, the Fungal Diversity Survey, is we're actually recording a lot of these and then sequencing them. And here we go.
Oh my God.
Gabrielle, here we go. This is the director of Fundus, I was just talking about. So she's in charge of helping sure we document all these mushrooms and we go ahead and sequence them. So what is this? What do we got here?
This one's not as rare. One of our
hold it up by your face. I want to just show people there.
It's huge.
Yeah, it's huge. Look at the thing.
Yeah, this is a Bondarzuia species. It's Bondarzuia berkeleyi down here in Missouri. And then in the West Coast, there's a rarer species called Bondarzuia.
Oh, I know. It's brown colored.
Ooh,
I've never found it. It's cool though. It's a Berkeley's poly. It's funny it's called Berkeley's polypore. It's not in Berkeley. It has nothing to do with Berkeley as far as I know.
Occidentalis.
Which means west.
Yes,
yeah, the west version.
Slightly different species, and it's a bit more rare out there than here. But so these are great, beautiful specimens. A little bit dried up. Yeah.
I'm just trying to catch you so we can hear everything.
Yeah,
That's so cool.
Bondarzuia.
What an amazing mushroom. All right, so we're gonna keep moving down the line here. We got a couple of dried out Lactarius and Russula, and the Berkeley's polypore here is actually in the Russula order. So it's a big fleshy polypore, but it's actually related to brittle-gills and Russula. And that's again the way that things evolved on that big chart I showed you right at the start. So here's some stinking mock oyster, Phylotopsis nidulans. This stuff, it smells like farts. It looks like it should be edible. Whoops. Oh, sorry. My gimbal is a little unhappy with me there. I think we're unbalanced, so give me 1 second. I'm going to get this rebalanced. So the stinking mock oyster is it's really smelly. It just smells like sewer gas and farts. And it's pretty funny. All right, this is going to try to reorient, and I'm going to tell it not to do that. Okay. Well, my gimbal is fighting me here, so I'm just going to take you guys off the gimbal, and we're going to keep going. So I apologize if this becomes a little bit shakier than it was. but the gimbal is giving me technical issues. So here you can see the whole room. Here's a little bonus fungi. This is some sort of a Notus weeping polypore. This thing grows on oaks. Big, nasty, weird-looking polypore, but it'll often have a lot of gutatation droplets of liquid on it. So this is one of my favorite ones to find and photograph. This isn't like a super pretty specimen, but it's pretty cool. So yeah, we're gonna go back to the table. We'll keep moving down here. Let's see, what else we got around? So this is Pleurotus austriatus. So this is just an oyster mushroom, pretty standard. As we come along here, we get into agarix. So we have, these are Pholiota. These are like wood decay, slightly parasitic mushrooms. These are pretty cool. These will glow under UV, and actually I can show you guys what that looks like. There's some mushrooms that are UV reactive. So I have a little UV light. I got this on Amazon. It's a 365 wavelength. And this mushroom will shine back. So there's compounds in the mushrooms that when I shine this light on them, the electrons get excited, and then as the electrons go down in energy state, they emit a light at a different wavelength. And that's what fluorescence is. So these mushrooms are UV reactive. There's some, like these oyster mushrooms don't really react much. These ones, Pluteus don't. I know some of these Russellas will. Some Russellas will sort of like react. Their gills are a little bit bright. Yeah, Berkeley's polypore is doing it a bit. I'll find Alan Rockefeller before he leaves. Hey, Alan, where are you going? Want to say hi to YouTube?
Hi, YouTube.
This is Alan Rockefeller. He's a pretty damn well-known mycologist, and he's got a fancy UV light which he can shine on things. It's very bright. Is there anything around that's really reactive? You want to show us?
Looking for Cortinarians.
Oh, some quartz, huh?
So those that glow in the dark, but they are not fluorescent at all.
Yeah.
And then sometimes just like the veil on these honey mushrooms
that's
glows.
cool.
So that's kind of cool. But there's a lichen over there that's really good.
Oh, yeah?
Oh, this is kind of nice.
Oh, that's a good one. Yeah, that's doing it. That's doing the thing. I missed the, God, was it the bolete that grew on the earth balls we saw? It was like, kind of glowed like orange. Remember that one? That was a pretty cool one.
Pseudoboletus parasiticus.
Yeah. And so we have some
Foliotas.
Yeah, those. Yeah, I was showing them with my light, but it was like your light looks way better.
It's pretty bright.
Mm-hmm. That's great. It's exciting.
But I had a really
here.
bright Cortinarius, but I don't know if I figured it out.
Oh, I saw that last night. Yeah, it was the gills were like super reactive.
Yeah.
Oh, that's cool.
What they're doing
I don't know if there's a Cortinarius section out here.
Yeah.
Let's look at that lichen.
It's around. Yeah, show me. I just figured I'd do a live stream to try to show everyone the stuff that was here. You know, it's like so cool. Oh yeah. Okay, so we found this last night, and this stuff just grows super bright
down
yellow.
your exposure compensation.
Yeah,
Some,
yeah.
So it's not just blown out, but
I can't do much on a live stream, but yeah, there you go.
Oh yeah, it's got pretty good structure. I feel
Yeah,
like.
it looks really.
Yeah,
so
let's
this is
do a
like
macro
so
shot of
take it away. So this stuff's green, and then when Alan puts the UV light on it, it glows like bright yellow. Just absolutely amazing. So you see that there's a lot of compounds and lichens that will fluoresce, and some of them are like chlorophyll, but there's a lot of other bioactive compounds and lichens that will glow different colors, and so it's really cool to like look around and see.
This one got identified by the lichen expert Natalie. She was giving the lichen talk, so it's Pyxine subcinerea.
Oh yeah, we have like world-renowned, expert ID people here. So like when you bring a sample in, you have people to tell you what it is. We're gonna like catalog the DNA. We're gonna add it to these surveys of things.
That's pretty good.
That one's pretty good. Yeah, that's bright. It looks. We put it off slightly to the side. It's easier to see the fluorescence when it's not like directly on it. Oh yes, some people are saying hello, Alan. So you are known. So yes, here we go. We got like nerds with flashlights and a little bit of lichen. Look at that. Oh yeah, that's really cool. So again, this is green, but it's glowing like bright ass chrome yellow. So really cool to like be able to differentiate lichens and stuff like this. So let's see, we have a couple other cool mushrooms here, things that I have never seen. Oh yeah,
Have you got a UV light?
I do.
Any chance you can just show in the jack-o'-lantern?
It's not going to glow. It glows in the dark. It's bioluminescent, but it's not UV reactive.
Okay.
That's different. If you follow Alan around, he's looking at all the things that are UV reactive, though. So he'll show you. So this is cool. This is a microheterotroph. So this is a plant that grows on fungi, derives its carbon from the mycelial network. It was originally thought as being a parasite. I think it's a pine sap, maybe. Good. This isn't I guess it's Monotropa uniflora. One of these is a pine sap though, and it's a different species. It's like Monotropa hypo something or other. So these plants are parasitic on the mushroom mycelium. They can derive all their carbon from the mycelium, which is it's a really cool lifestyle. So here's a Caloboletus. This bolete's mildly UV reactive. This is super bitter, inedible bolete, but they're interesting looking. This is a big, weird, funky, beautiful polypore. I'm gonna give you guys a little thumb tap on this thing. This is just like people always ask me, like, the jokes like, oh, I can eat every mushroom once, ha ha ha. I was like, I dare you to try to eat this, right? This is like hard as wood. You know, I could take a big bite of this and I would go nowhere. So, no, you can't eat every mushroom once. There's just some mushrooms you can't eat. It's a big piece of wood. So, moving down the line here, we got a lot of, like, polypores. I'm going to keep my UV light out, see if anything's reactive. What is this? Pseudoanotus. Okay, so this is the same weeping polypore thing I was looking at earlier. Let's see what we got here. More Pseudoanotus. Lots of different polypores and crusts. Like I said, it's been kind of dry, so we were, not finding super prime specimens. But here's some nice Latiferous sulphurus, chicken of the woods. We have a more mature specimen. So this is a really young one. Here's a more mature one. Occasionally you can see chicken of the woods will be slightly UV reactive on the pore surface. A lot of polypores, their pore surface is somewhat reactive to UV. Not like crazy, but a little bit. Here was some maitake. Me and my friend Mandy found this one the other day, Mushroom Madman. She's the reason that I came to NAMA, because she hit me up and was like, yo, you should come to NAMA. And I was like, I haven't been to a mushroom festival this year. Sure, I'll come to Missouri. I've never been. And I am terrible at spotting maitake, but she's really, really good at finding them. So Mushroom Madman, Mandy Cork, found this beautiful maitake, and it's here on the table, which is good to see. Let's see, here's some Ganodermas. Oh, so here's the whole sequence of chicken woods. We have sort of a nascent, immature one. It's grown up. It's still pretty soft. We have this big kind of one that's like starting to get dried on the edge. And here's an old one. This is completely inedible. It's totally dry. You could try powdering it, but at this point, it's better to just leave it. So let's see. We got some Ganodermas. This is like probably an artist conch. This is an really old pheasant back, Stereoporus. Look at those pores on there. I hope no one has trypophobia. Fear of tiny irregular holes. Yeah. Okay. Let's see. This is Royoporus badius. We get a lot of this in California, but we just call it the bay polypore. Here it's grown on a different substrate. Pretty common around the world, though. You see it all over the place. Oh, here we go. Here's some Postia. Little polypores. These smell really weird. They have a very kind of interesting, like fruity, almost like chemically aroma. Here's this cinnabar bracket. Oh, these are so cool. Look at that beautiful orange polypore. These probably have a pretty cool reaction to KOH, potassium hydroxide. If you guys hang around, I'll show you some of that. Let's see, we got Trametes betulinum here. So this is related to a turkey tail, but instead of having little pores, it has little gills. There's still a polypore mushroom, even though they have gills, and that's confusing. But evolutionarily, like I showed you guys, things evolve from one another, and so this is called convergent evolution, where this kind of mushroom evolved little pores, and then went back to having gills because that's an effective strategy for spreading spores. So come along here. This is the actual turkey tail. It's also a Trametes species, so it's related, but it has little pores underneath instead of having those gills. So understanding the difference here between like Trametes versicolor, which has little pores, versus Trametes betulinum, which has these gills, helps you identify and understand mushrooms. So really cool to have all these things laid out and ID'd by experts, and it's just an amazing learning opportunity to be here. So we're getting into Hericiums. These are all tooth fungi morphologically, but Hericiums are actually in the Russula family, so similar to the Berkeley polypore that I showed you earlier, and these are related to Russulas and boletes and Lactarius, and they're part of that evolutionary lineage. But lion's mane is a great edible mushroom. It's all these cool like bioactive sesquiterpenes and polysaccharides and things that people say will help improve your cognitive function. Someone gave a talk earlier saying that there's a lion's mane sort of smoothie that someone was using to actually reverse Parkinson's. I'm still kind of skeptical of that, but it's a really, if nothing else, it's a delicious mushroom. It's incredible to cultivate. This is Hericium. Actually, this is cool because we have multiple different species of Hericium. There is lion's mane, the Hericium erinaceus. So this is kind of the one that you see all the time cultivated. Then there is Hericium coralloides, which has a more branched, open growth pattern. And there's somewhere here there was this is it, Hericium americanum. So this is what's called the bear's tooth. And it looks very, very similar to lion's mane. I'm not totally sure how to differentiate them other than probably doing microscopy, but people here know what they're doing, so I trust them. Here's a crazy tooth fungus. So this is called a respunate tooth fungus. So it's growing here on wood, but it has all these crazy teeth underneath. So these things aren't clustered genetically, they're clustered because they look similar. They have these teeth, right? And so that's one of the big differences is like mycology is this kind of big trade-off between like, do they look similar or are they genetically related? And all of mycology used to be just based on how things looked. But because we have access to DNA technology and we can sequence things, we become aware of these relationships that although these things look similar, they might actually be related. This is more closely related to like this Berkeley's polypore or a Russula than it would be related to all of the other polypores or even the other toothed mushrooms. Like these look sort of similar, but they're not related. And that's cool because it's that example of convergent evolution, things converging on similar traits because it's effective. There's only so many ways to spread spores or something like that. So here's a bunch of, these are called Apioperdon, little pear-shaped puffballs. Apioperdon pyriform. So I added at least a couple of these to the table. I'll give these a puff, but I don't really want to put spores all over the place. Here's some of the cool, this is a really weird looking, interesting crust. That's fun. Here's some club and coral fungi. I know I put a couple of Romaria on the table. I think this is my specimen I brought in the other day. So these are they're called coral mushrooms, but Romaria, claviloid. So those are cool. Those are completely different standard from, not a cap and stem kind of mushroom. But these are still in Basidiomycota. We'll get into the Ascomycetes in a little bit. Then we have some wood ear fungus. You see this a lot in Chinese food. Pretty much any spring roll, egg roll kind of thing you've had in a Chinese restaurant has some wood ear fungus in it. Okay, we're going to move on to Ascomycota. So everything we've been looking at so far has been Basidiomycota. Here's Ascomycota. Actually, these are slime molds. So let's see. What is this stuff? This is Fuligoseptica. So this is dog vomit slime mold. It's usually very yellow. Right now it's kind of old. Here's some wolf's milk slime mold. So when these are young, these are pink, and you can pop them with your finger like a little zit. We're gonna get into Ascos. So, here's this. Someone found this today, and it's just like the coolest, weirdest yellow Ascomycete. I've never seen anything like it. And it's full of like jelly inside. I'm just blown away by this thing. It is. I know it doesn't look like that much to you guys, but to me, this is utterly amazing. And it is Entomonema liquescens. And yeah, it's full of like a weird jelly stuff inside. And it looks so different from most Ascomycetes. A lot of Ascomycetes live as what's called endophytes. So in their asexual form, which means when they have one copy of their genome, they live inside of plants. And they live in between plant cells as part of like the plant microbiome. But they can usually live as a mutualist or at least commensalist, and then they're not really affecting the plant. But occasionally, when conditions shift with its humidity, temperature, nutrient availability, whatever, they decide to kind of become pathogenic and infect the plant with like a disease. And the plant will be like, well, that noise, and drop the leaf. It's kind of like, you have like a rowdy tenant in your building, you kick them out. So the plant is the landlord and the endophytes are the tenants. And that's what some of these leaves are examples of, like a thing that became parasitic, but it was an Ascomycete that was probably just living as part of the plant microbiome until things kind of shifted. This is, what is this, the green elf cup, the Chlorosebaria. And right now it's not forming the little green cups, but its mycelium stains the wood this beautiful kind of, green color and has a little sheen to it. There's a guy on TikTok, Justin Trees, who talks about chatoyancy, and it's like the sheen on wood. And he says this green wood is like really highly valued by woodworkers. It's not super solid, so you don't want to do anything structural with it, but it's really great for ornamentation and woodworking. So Chloris Abaria, the green elf cup, is a really beautiful, pretty common Ascomycete cup. Someone picked these up. This is another really tiny, cool Ascomycete cup fungi. And it's, you go anywhere in the world and you pick up a wet piece of wood or kneel down on a log, you'll probably find some ascos on it somewhere, maybe even a slime mold. But we had to hunt pretty hard for this kind of stuff today because there was just not a lot out there. Here's more of that weird yellow asco thing. Here's some lobster mushrooms. So these are cool. This is a base species of Russula or Lactarius that gets infected with Hypomyces lactiflorum, which turns it sort of an uninteresting white mushroom into this bright orangey yellow mushroom that's actually a very good edible. So it's pretty cool to see Hypomyces. But this is an example of like a Basidiomycete mushroom that gets parasitized and colonized by this Ascomycete mold. And it completely changes like the flavor and texture and gene expression of the mushroom below and turns what would have been a mushroom to produce basidiospores into this sort of mold-producing spore factory for the Ascomycete. So that's really cool. There's a few more dried out things. We go to lichens. So most lichens are actually Ascomycetes, but lichen is such a complex, interesting form of life, and I've been thinking so much about it while I've been here, because lichen is a combination of fungi and algae, and either it's algae or cyanobacteria, but it can be either one. It's usually what's called a mycobiont. So the fungi forms the structure, the algae does the photosynthesis, and then there's a whole bunch of yeast and bacteria that actually live inside of the body of the algae, inside the body of the lichen, and sort of do like, let's call it translation between like the algae, the mycobiont, and the photobiont. So it's a whole ecosystem unto itself. And lichens are all over the planet. There's tons of diversity, and you can chop and change the different kinds of fungi and algae and cyanobacteria and yeast and bacteria inside them, and they'll end up looking totally different and be able to grow in totally different environments. So there's just this incredible, untapped, diversity of lichens that we are just starting to kind of understand. The other crazy thing that happens is because it's all these yeast and bacteria and fungi and algae all living together, is there's this constant chemical chatter within. And because of that, it means that lichens are usually filled with really potent, interesting bioactive compounds which are being explored for like drug discovery kind of stuff. So that's pretty cool. And here's a great little life cycle diagram for fungi. And this is a great one. Usually we start like a spore, and it germinates. And it starts forming hypha, which are fungal cells. And then two hypha from their one end. So it has one copy of the genome. Two hyphae will fuse in a process called plasmogy, and it will form this dikaryote. It has two versions of the genome. Humans have two N. Our sperm and eggs have one N. So that's kind of how this works. We went from one N to two N after two hyphae fused, and then it undergoes mitosis, which doubles, and you're getting formation of a mushroom, and then. Let's see, we go from diploid to 2N, we do meiosis, which breaks us back down to spores, and we become 1N again. And then it forms these spores on a basidia. So this is how Basidiomycetes work. Ascomycetes are similar, it's just the spores look differently, they're packaged differently. Basidia is kind of like a club-like thing with all the spores in the end, and this is what hangs down underneath the gills of a mushroom or in a polypore. And then distributes the spores, and the cycle starts all over again. So there's fungal things. Here's some basics on, fungal identification. The cap. Look for textures on the cap, scales, warts. Look at the gills. Look how the gills are attached. We can talk about different forms of like how caps look, and if there's a vulva or not, how the gills are attached, different sort of shapes of mushrooms. Thinking again about the ring or an annulus, some sort of skirt, partial veil underneath, the stem or the stipe, the base of the mushroom, and there's a cup or a vulva or anything around it, will tell you what kind of mushroom it is. Life cycle, we just have all these cool resources here about gills and stem and stalk, and it's great to sort of have common names and science names for all these things. Oh, here's Mandy. Say hi. So she found the great maitake I was talking about earlier. She's the one who convinced me to come. So. And you are now what, the NAMA 4A leader? Yeah, there you go. Yeah, she's proud of herself. It's good. Good stuff. Yeah, if you guys want to come to NAMA and experience all this cool stuff, all these nerdy people and all these amazing mushrooms and, stuff. Mandy's your gal. She's going to be playing it all next year. So.
Hey. Yeah, I'm going to go back over there. I'm just letting you know.
Cool. Yeah, I'll see you over there in a sec. So we just have all these cool things. This is the heartbeat of this whole place. These are the people who are sitting here cataloging these specimens, getting them ready for sequencing. Say hello. We're streaming on YouTube, so I'll just put this up later, and you guys can. So all these things have been sort of pulled aside, and they're actually going to do DNA sequencing on these things. And so these are what are called eyelash cups. They are an Ascomycete cup. So it's just really cool to see all this stuff. So there's Alan teaching people. Everybody learns from Alan. It's great to have him here. And here's some more tables of stuff that haven't been identified yet. There's, this is a process. Things come in, people say which foray they got them on, they, sort of put them out, and then experts come by and start cataloging things, connecting them to observations in iNaturalist, and then we're making decisions about to voucher them, to sequence them. There's Stephen Russell. Say hi, Stephen. So he's doing a lot of our sequencing. You were at Purdue. Purdue, I guess. But there's Jacob. He knows everything about mushrooms. Here's just, John. John has been doing all the photography. These are all members of NAMA, all people who are doing really important stuff with mushrooms, and just, it's a little dry, so we got a lot of things on sticks and crusts and dried out mushrooms, but there's just a phenomenal diversity of different mushrooms out here, which is really cool to see. So, is this the elm oyster? What is this? Hey Jacob, is this the elm oyster, like the veiled oyster? Is this a tricholoma? What is this? Yeah, it's the elm oyster. Yeah, pretty. Can you give that a little slap for me? You just get a little
like just the cat.
Yeah, just give me. Yeah. Give me another one of those. I like that. Yeah. See, it's a big sturdy. So oysters don't have stems. Well, oysters do have stems, and this one in particular has a big fatty stem. Yeah. You'll find it growing out of dead wood, usually elm trees often, but not always. So these are, morphologically, would we say this is like the North American version of like a king trumpet almost? Could be, sort of, it's not really. It looks different, but ecologically, sort of speaking in terms of the organization of oysters, maybe this would be sort of analogous to our North American version of the Pleurotus syringae. That's how I've thought about it.
They're in different suborders.
They're not related, but I'm just thinking about the size and the depth of it kind of thing. So. It's been a hell of a weekend, and earlier they're doing all sorts of microscopy workshops, and over there they're drying mushrooms out, getting them ready for sequencing. Steve Russell was doing a whole workshop today on running people through the DNA sequencing and how to do that process, sample preparation. Here's a bag of leftover junk that we'll throw back out in the woods. Here's the camera setup that Gabriella from Fundus has been using to document and catalog everything. So we're taking really nice macro shots, having them on the computer. We're linking them up with the voucher numbers and the accession numbers. As always, mycology requires a good deal of decent whiskey to get through a whole bunch of mushrooms. Let's go check out what Alan's doing. I see there's people always clustered around him, and he's always doing interesting things. So. What do we got here?
I haven't seen the SVO before. I have an SVO.
Sorry. Alan, are these you reactive things? You want to show me what we got?
No. We're actually just comparing the lights.
No. Yeah. Oh, this is cool. Oh, wow! Look at that! Holy cow! What is it? Is this Hypholoma?
Mycena inclinata.
Oh, okay. Neat. Yeah. That really glows well. It shows up well on camera,
pretty,
too.
That's so beautiful.
Kind of bright at the stem base.
Mm-hmm. Yeah, it's different how it changes across different parts of the mushroom, much more purple there, kind of more yellowy there. Yeah, I mean, most of these UV things are hard to see on camera, but that one shows up really well.
Yeah.
That's great.
Another really pretty one is Atheniella because it has these rhizomorphs at the base, and only the rhizomorphs are fluorescent.
Neat.
That's gorgeous.
Wow.
Yeah.
So, what a cool haul of mushrooms. So, I'll kind of run you guys down the line here just one more time of looking at everything. Excuse me, guys. So, here's our chanterelles and trumpets, canthorales kind of thing. So these are dried out black trumpets, craterellus. Here's chanterelle. Here's these little like cinnabar chanterelles. Some smooth chanterelles. Go back to foliotas. What do we got here? We got the stinking oyster. We have the pleurotus, and the pluteroid type mushrooms. We got the big old Berkeley's polypore and the russelas. I'm getting some amanitas here, some tiny little things, honey mushrooms, jack-o'-lanterns, Lacaria. This is again based on like spore color and some other morphological characteristics. Here's a clavelina, or rumeria, and claveloid mushrooms, little coral things, rumeria. All of our puffballs, Apioperdon, some crust fungi, some tooth fungi. It's crazy, like, lion's mane, great lion's mane, the Respinate. No, you're fine. Isn't that cool? Like, yeah. I was just talking about how it's so cool that like we have all these all these like crusty things, toothy things. None of them are related necessarily. But they have that similarity, and everything is like agreed that okay, this seems like a good strategy to disperse our spores. You've got that, you've got this, all these different ways of doing business.
Incredible.
Yeah, and it's cool now because science used to think that these were all related, but now we know better. But they still look cool, and we still group them in this way because it makes sense to us, from an eye sense. But yeah, genetically they're all different. So.
They know the truth.
They know the truth, yep. Now we're starting to learn. We're just starting to learn the truth about fungi. There's just so much to learn. So yeah, all these polypores, a big chicken of the woods. We have the maitake, so this is hen of the woods, chicken of the woods. People always get those confused, but they are different. This is Latipris sulphurus, and that is Gryphla frondosa, and so that's why it's good to sometimes always use both the science name and a common name, because then it helps people not get confused. These are the pseudoanotus, the dryadius, the weeping polypores. Here's this big old crazy lump of polypores that no one can eat this, right? People are like, oh, I'm gonna eat every mushroom once. Yeah, dare you, try. Take a big bite, you're not gonna get anywhere. That's it. We have not many boletes because it's been dry, but there's an old man of the woods, Strobillomyces, like a goth mushroom, It's not black because it's old, it's black to begin with. It is old and a little dried out, but it's also black, even when it's young and fresh. So, and here's the microheterotrophs, these Monotropa uniflora and the other Monotropa species. Yeah, it's been really cool being here at NAMA. I'd highly recommend anyone who's into mushrooms register. I think the foray next year is going to be in Asheville, North Carolina. It's going to be really exciting. My friend Mandy, who I showed you guys, is going to be in charge of setting up all the forays and different things like that. So if you are interested in mycology, if you are a science person, if you're a nature person, if you're just want to hang out with some other nerds and learn about mushrooms, is a really, really cool place to come. So check out the North American Mycology Association, nama.org. There's also an Instagram. Check out the Fungal Diversity Survey, Fundus. They're here on YouTube. Check out and use iNaturalist. That's the #1 app that we're all uploading our photos to. It's got a really good automated algorithm. You can take a photo of a mushroom, upload it, and it'll spit back an idea of what that mushroom is, and there's experts who can come along and help verify your identification. Once your identification is verified, then scientists can actually use that to help track species distribution. People are using it to track climate change, all sorts of stuff like that. So there's some really cool things that are happening here, and it's just fun to be here with a bunch of mushroom nerds. So I hope you guys have enjoyed this live stream. Maybe you learned something. Share it with your friends, share it with your family. If you want to learn more from me, Gordon, Fascinated by Fungi. I got a website, fascinatedbyfungi.com. All my links are on there. I'm on every imaginable social media platform, YouTube, Facebook, Instagram, TikTok, etc. I have a Patreon if you want to support me. I have a website. I have cool t-shirts and original mushroom designs. You can get stuff. I also have a podcast. Fascinated by Fungi is on Spotify, Apple, Google, Callin, here on YouTube too. So anyhow, check it out. Yeah, thank you for coming along for the ride. Everyone have a good night and sending you all much love. So, bye!